Hello everyone, this is Dr. Abhay Vikram Singh, Department of Orthopedics. My topic is peripheral nerve injuries. And so this in the slide, we can see the structure of the nerve, peripheral nerve. In this, we can see anatomy of the peripheral nerve, the longitudinal vessels, the outer epineurium, and the inner epineurium we can see. And we have myelinated nerve fibers and unmyelinated nerve fibers. There are two types of nerve fibers, myelinated and unmyelinated. There are several structures in the nerves. They are neuron, giant cells, node of Ranvier, nerve fibers, and fascicles. About neuron, it's a nerve cell made up of cell body in dorsal root ganglion for afferent fibers and ventral horn for efferent fibers. Dendrites, they receive signals, and axons, they transmit signals. The giant cells are Schwann cells produce myelin to cover axons. Myelin increases conduction speed. The node of Ranvier or gap between Schwann cells facilitates conduction of action potential and impulsive signals. Nerve fibers, a single axon. There are three types. Large myelinated fibers are fast. Small unmyelinated fibers are slow. Efferent fibers that are called axons transmits motor signals from CNS via ventral horn to peripheral muscles. Afferent fibers, axons, transmit sensory signals from peripheral receptors via DRG to CNS. Fascicles is a group of nerve fibers surrounded by perineurium. Fascicles unite and divide from plexi, then continuously along the course of the nerve. The next structures are peripheral nerves. One or more fascicles surrounded by epineurium. Most peripheral nerves have both motor and sensory fascicles. Epineurium surroundings all fascicles of peripheral nerves, protects and nourishes fascicles. The perineurium is surrounds individual fascicles, protects protects and nourishes nerve fibers. Endoneurium is intrinsic vascular plexus with the endoneurium, perineurium, and perineurium. The physiology of membrane events. Action potential generates depending on an attribute that the axon shares with other excitable cells, the resting membrane potential. This is a study potential difference across the neurolemma with the inside of the fiber electronegative with respect to the outside environment. This potential is generated by the difference in concentration of certain ions between the inside of the neurolemma and the outside. The most important ions involved are sodium and potassium. A concentration gradient develops in response to the differential permeability of the membrane to the ions, which allows the potassium that is present in high concentration within the cell to move down its concentration gradient to outside. Negative charge within the cell is primarily mediated by large molecules such as proteins and amino acids that are not diffusible to the membrane. Thus, the potassium moves the membrane until the outward force generated by its concentrated gradients is precisely counterbalanced by the inward force produced by the developing negativity left inside the cell. The electric potential required to exert this restraint is called the equilibrium potential for potassium, and it can be calculated from the ionic concentration on the two sides of the membrane by the formula known as Nernst equation. This can be derived from the basic principles of thermodynamics, and for practical purposes, it is exposed as E equals to K log C1 by C2, where E is the equilibrium potential in millivolts, and C1 and C2 are concentrations of the ions on the outside and inside of the membrane respectively. K is constant resulting from the thermodynamic derivation of the equation. The equilibrium potential for potassium is about minus 88 millivolts with the inside negative relative to the outside. Since sodium is present in higher concentration outside than inside the membrane, its equilibrium potential is about 75 millivolts. That is, the inside of the cell would have to be 75 millivolts positive relative to the outside to prevent invert diffusion of sodium along its concentration gradient. In the resting state, however, the membrane is much more permeable to potassium than to sodium. Thus, the resting membrane potential is about minus 80 millivolts, which is more closer to the potassium than the sodium equilibrium potential. The fact that this value is slightly more positive than the potassium equilibrium potential reflects the fact that there is a small invert leakage of sodium that slightly discharges the potential differences. In a series of landmark papers in 1952, Hodgkins and Huxley postulated that it is this difference permeability to potassium and sodium and the resultant resting membrane potential that provides for the axon's ability to generate action potential. When an electric current is applied to the axon, a small depolarization develops cathodal pole of the electrode, that is, the inferior of the cell becomes slightly less negative relative to the outside. If the depolarization is great enough and occurs over the relatively short period of time, the axon reaches threshold. When this occurs, there is dramatic increase in the membrane permeability for sodium. Since the resting state, both the electrical and the chemical gradient strongly favors sodium influx, there is rapid explosive inflow of the ions down to its electrochemical gradient. This moves the membrane away from the potassium equilibrium potential towards the sodium potential. The membrane potential then moves from being strongly negative towards zero, and actually the inside of the cell becomes positive. That is, it is an overshoot. This rapid influx of sodium soon ceases. The membrane permeability to sodium quickly turns off. The potassium permeability increases and is again predominant. Since the interior of the cell is now much more positive than the potassium equilibrium potential, this ion is expelled in response to its electrical gradient, removing positive charges and consequently repolarizing the membrane. The end result of this entire process is to leave the cell interior with a slight increase in sodium ions and a corresponding decrease in the potassium ions. Although the change in the ionic balance produced by a single action potential is extremely small, it particularly discharges the resting membrane potential, and it left unchecked would eventually depolarize the membrane, making the cell inexcitable. The proper internal external sodium potassium relationship is continuously restored the action of the sodium pump. This is the membrane bound enzyme that operates via an active process utilizing energy liberated by cleaving the high energy phosphates bond in adenosine triphosphates. The enzymes is a sodium potassium ATPase that extrudes sodium from inside the fiber in exchange for potassium, which is pumped into the fiber, thereby restoring proper ionic balance. It is important to emphasize the fact that this is an active process requiring the use of metabolic energy as opposed to the purely passive events of the action potential that occurs in response to the electrochemical gradient across the membrane. Furthermore, the activity of the sodium pump is a housekeeping function, and it does not participate in the rapid potential changes that constitute the passive of the action potential. In fact, for a large axon, Many thousands of action potential can be still elicited without a dramatic decrease in resting membrane potential after the sodium pump is poisoned with metabolic blocking agents. Once the action potential is generated, it must be conducted along the action. Upon development of an action potential, the interior of the membrane in the local area has an excess of positive relation both to be exterior of the membrane at that point and to neighboring exoplasm inside the cell. This generates a local current flow that spreads down the axon in uh, according with the cable properties of the nerve fiber. This term refers passive electrical properties of the axon. The net effect of this local current is to depolarize the adjacent segment of the membrane so that it is in turn reaches the threshold necessary to generate another action potential, beginning in the process again and continuing to the length of the fiber. Thus, the action potential is conducted along by the axon by a sort of sequential depolarization and regeneration. The process has been a term on and all non term and all or none phenomenon, referring to the fact that if depolarization does not cross the requisite threshold, no potential is generated. But when this value is achieved, the potential is developed with equal amplitude along with the entire length and doesn't decrement during conduction. From the above discussion, it will be realized that if each tiny membrane segment had to be sequentially depolarized for conduction to occur, this would be a very slow process indeed. According to the laws of the cable theory, conduction velocity increases in proportion to the square root of fiber diameter, and thus faster conduction develops with an increase in the size of axon. This is seen phylogenetically in lower animals, and the process reaches in zenith in the squid, where some of the axon of mantle nerves are 0.5 millimeter or more in diameter. Increasing conduction velocity by axon size is an inefficient process, and the conduction velocities necessary for functioning of more advanced species would not be possible unless a different method had evolved. Higher animals, therefore, have developed the myelin sheet that insulated the fiber. As noted above, the sheet is laid down by Schwann cells that are linearly adjacent to each other along the axon. At the junction between the cells, there is a small gap that is membrane is exposed to the surrounding extracellular fluid. It is at the gaps that the nodes of Ranvier that the generation of the action potential occurs. In these myelinated fibers, the local current that develops an excitation spread along the axons of depolarization, the membrane at the node instead of the next immediately adjacent membrane site. This allows the action potential to jump from one node to node of Ranvier to the next in the phenomenon known as saltatory conduction. This greatly increases the speed in transmission and provides for the rapid conduction velocity necessary in the nervous system of higher animals. It has been demonstrated that the axon has very little synthetic capability, and thus the proteins and polypeptides necessary for maintenance of the neurolemma and internal organelles must be supplied from cell soma. Furthermore, furthermore, the substances expelled from the distal axonal terminals that are necessary for neurotransmission or for the tropic control of muscle fibers or other nerve cells must also be supplied by the perikaryon. The mechanism underlying the supply system has been largely elucidated in the past 10 years and is known as axonal or exoplasmic transport. Theories of material flow in nerve date to the pericrystal era, But progressively, more attention has been paid to this phenomenon since the work of Weiss and Hisco in 1984, which appeared to show accumulation of exoplasm proximal to the ligature placed above the nerve. They concluded that the exoplasm actually moved as bulk flow in the proximal distal direction. Subsequent work by many investigators has revealed that this concept is probably not correct, but that in fact there is transport within the axon as a number of different species substances synthesized in the cell body and necessary to function the axon and effectors organs. The process appears to be remarkably uniform among fibers of different types and has been shown to move at the same rate and uh, various sized myelinated and unmyelinated fibers as well as in the autonomic nervous system. The synthesized materials consist largely of proteins and polypeptides and can be tagged by injecting the radioactively labeled precursors amino acids such as leucine into the region of the neuronal cell bodies. Example, into the spinal dorsal root ganglion or ventral horns of cat. The precursors is then rapidly within 15 to 20 minutes synthesized into the proteins and polypeptides and exported into the axons of the transport distally. The rate at which the substance move can then be calculated from the time of appearance of the radioactive in the axon and its movement distally. This rate has been determined to be about 410 millimeter per day and is remarkably uniform among various species. Much of the work has been done in cat, but when various animals, including frogs, dogs, garfishes, and primates have been studied, and the data corrected to mammalian body temperature, very similar result have been obtained. Fast exoplasmic transport can be studied in vitro by injecting the dorsal root ganglion of the seventh lumbar segment in the cat with leucine and then removing the entire sciatic nerve complex, including the dorsal roots and ganglia proximally and the peroneal and tibial branches distally. A period of about 2 hours is allowed between the time of injection and removal of the nerve from animal to permit incorporation of the amino acid precursor or into proteins of and polypeptides and their subsequent from the cell body into the axon. The nerve is placed in the environment in which the transport will continue. In practice, two such systems are employed. The nerve may be placed in a flask of pre-oxygenated Ringer's solutions to which can be added various agents whose effect or on transport can be assessed. Exoplasmic transport also continues if the nerve is placed in a sealed metal chamber in which the temperature and humidity are controlled, and this device permits electrical stimulation of the nerve and the continuous monitoring of the resulting action potential. When the experiment is concluded with either system, the nerve is then removed and segmentally assayed for radioactivity to allow the computation of flow rates. In this way, the effect of various experiment parameters on the characteristics of exoplasmic transport can be assessed. Much of the work has been done in cat, but when various with this method, it has been demonstrated that if the oxygen in the atmosphere is replaced with nitrogen, or if the nerve is exposed to metabolic prisons such as cyanide transport, it would take cyanide transport within about 15 minutes. If the one measure of the concentration of high energy phosphate in the nerve is, it is noted to fall approximately 50% of the control value of the time when exoplasmic stops. Coincidentally, action potential conduction ceases at about the same time. Agents that interfere with glycolysis or or citric acid cycle also interfere with transport, although time delays is somewhat longer because of the ability of the nerve to utilize metabolites other than glucose. All of the data are consistent with the hypothesis of the exoplasmic transport is an active process requiring the expenditure of the metabolic energy. Furthermore, this energy must be available segmentally all along the axon because transport is blocked locally if the small segment of the nerve is covered with paraffin to prevent oxidation. Thus, high energy phosphate apparently are not capable of diffusing from oxygenated segments into adjacent hypoxic areas. Although most experimental work has concentrated on proximal distal flow, it has also been demonstrated uh, that there is a retrograde system capable of transferring substances from the periphery towards the neural soma. It has been postulated that interference with this mechanism is a signal for the chromatolysis that occurs in the cell soma when the nerve is injured. Much investigation effort has been expanded in elucidating the mechanism by which the variety of substances might be transported in a nerve of different diameters at the same fast rate. One important clue was the discovery that agents that impair the function of microtubules can alter exoplasmic transport. Microtubules are linearly arranged organelles with a diameter of approximately 25 nanometer, such that are present in many types of cells and underlie such vital functions as mitosis. They have been shown to extend through, but the entire length of the axon are composed of individual protein dimer subunits called tubulin. It appears that there is a dynamic equilibrium between the soluble tubulin present in the cytoplasm and that which is assembled into microtubules. Certain chemical agents are capable of altering this equilibrium. Amongst all these, colchicine, which precipitates the tuberculin and has been useful in the experimental purification of the substance. Perhaps the most potent of these agents, however, are the vinca alkaloids, widely known in clinical medicine for their anti-mitotic effect in chemotherapy of various malignancies. It has been shown that the use of these drugs in concentration as low as 1 micrometer can block exoplasmic transport. Furthermore, uh, more this effect appears related to the loss of microtubular integrity. Interestingly, colchicine also interferes with exoplasmic transport. Consideration of the experimental data discussed above allows ones to construct a theoretical model of mechanism by which different substances can be bidirectionally transported with the axon. This has been termed the transport filament hypothesis in analogy of Huxley's sliding filament hypothesis for muscle concentration, and suggested various components are bound to transport filaments which in turn are moved along the stationary microtubules such as boxcar along a railroad track. The transport filaments are moved by the action of cross bridges, which utilizes the energy to restore the ATP. In apparent confirmation of this theory and relatively high level of calcium and magnesium, activated ATPase has been shown to be present in nerve fibers. Furthermore, nerve also contain a high level of high level of polypeptide calmodin, which is capable of activating the enzyme in the presence of micromolar levels of uh, calcium in the axons. This hypothesis then accounts for dependence of exoplasmic transport on local supply of metabolic energy and can also explain the sensitivity of system to agents that uncouple microtubules. From the above discussion, it might be anticipated that satisfactory function of the exoplasmic transport system would require an optimum concentration of various ions in the extracellular fluid and consequently inside the cell. This is the fact true, although the mechanism of studying this point is complicated by the fact that the perineurium act as a diffusion barrier uh, between two ions as noted above. Uh, it was found that one could not alter the extraaxonal environment simply by changing the ionic concentration in the medium bathing in the nerve. Therefore, a method was developed by which perineurium could be stripped of the cat perineural nerve under the operating microscope, allowing direct experiment manipulation of both extra and intracellular ionic environment. Using this technique, workers found that the concentration of calcium in the extracellular space must be maintained in the range normally normally found in vivo in order to for exoplasmic transport to continue. Magnesium is capable of lowering the calcium requirement somewhat, but cannot substitute it for entirely. These data suggest calcium experts a critical regulatory function, and that is binding to calmodin allows the later to activate calcium and magnesium ATPase present on the cross bridging mentioned above. Other ions may also be important, although their specific relation to the transport mechanism is more difficult to understand. The steroid alkaloid BTH has been shown to hold sodium channel opens, thereby uh, increasing intracellular sodium concentration. This produces depolarization of the cell membrane and also block exoplasmic transport in lower concentration. These experiments have been carried out in our laboratories and have demonstrated that uh, has been demonstrated that provide the rate of transport decrease by as much as 15%. Thus, two basic axonal functions of electrical condition and material transport are related related through alteration in the intracellular ionic environment.